here and bring up. Okay, what we're going to do again now is switch gears again. We've been talking about the molecules and how we can use these molecules in various tests. What we're going to do now is start addressing the question, well, what happens in an organism when an organism gets challenged with some sort of antigen? So we're going to look to see what happens in that animal. Look at the kinetics of the antibody response. All right, just as a reminder, we've told you all these things, but they've been sort of separated. But these are the main hallmarks of the immune response. I just want to remind you of self, non-self discrimination, memory, and that's what we're going to talk about today. We're going to talk about what this memory response is. I've been alluding to the fact that that when the immune response encounters the antigen the second time, it reacts differently. So we'll see what that difference is in the reaction. So what the memory response is. And, of course, specificity. There's a high degree of specificity in the immune system. All right. So let's first take a look at what happens to the immunogen. If we want to track what happens to the immunogen after it's injected into some organism or we get exposed to some sort of immunogen. All right, so we'll need to look, first of all, what happens to the immunogen after the first exposure, the primary exposure. What you see is if we were to inject, in say, an experimental system, a labeled antigen, something that we could track really easily as a function of time after injection, what you see is that initially there's a very rapid drop in the amount of antigen. So this is antigen circulating in the serum. And that's due to equilibration. What's just happening here is a physical thing. The antigen is being dispersed all over the body, and it'll equilibrate between the extravascular and intravascular compartments. And so you see this very rapid decline in the amount of circulating antibody or antigen. Then there's a more slow degradation or decrease in the amount of antigen, and that's called the catabolic decay phase, because what's happening here, the macrophages are beginning to metabolize this thing. We're breaking down this, and this is a metabolic process. It takes some time. But then roughly about a week to 10 days, somewhere in that ballpark, afterwards, you see a very rapid decline in the amount of antigen. That is the immune elimination phase. Because what's happened now, all this time between time 0 and about time 1 week roughly, the immune system has become primed, and now antibodies are going to be made. As soon as the antibodies are made, they complex with that antigen, and now you have immune complexes which can be very rapidly cleared from the system by the phagocytic cells. Now, if you see this antigen a second time, you get all of these three responses. The only difference is that the onset of this immune elimination phase is much faster. Instead of seeing it occur at a week to 10 days, you may see it occur after 3 or 4 days. So there's a more rapid onset of this immune elimination phase. All right. Now let's look what happens to the antibody. So we're going to follow the antibody response. And again, we're going to be looking after primary exposure. And since if you remember, I had told you that there's a difference in the response to T-dependent and T-independent antigens, we'll first discuss the T-dependent antigen and see what the response is, the primary response to a T-dependent antigen. All right, what you see initially after, and again, we're looking at antibody concentration here, antibody titer, days after immunization. If we administer the antigen here, for a while we don't see any antibodies. It takes about a week to 10 days. For most antigens, that's the ballpark you're talking about. Now, it varies a little bit from one antigen to another. Some antigens may take even, several weeks before we mount an immune response. But mostly, it's about a week to 10 days, somewhere in that range. So we don't see anything. That's as far as antibody is concerned. That is referred to as the lag phase. And in fact, all that clearance that we just talked about is occurring during this part. The immune elimination phase is going to occur right about there, where we start beginning to see antibody being produced. All right, so we have this lag phase. Then there's the log phase, because you're getting a logarithmic increase in the concentration of antibody. But eventually, remember, B cells differentiated into plasma cells. That plasma cell is the terminal stage in differentiation. Those plasma cells will start dying off. And eventually you reach a situation where the amount of antibody, new production of antibody equals the rate of antibody decay, and you enter into this plateau phase. And then finally, when there's no more antibody being made, all the plasma cells have died off, you just degrade the antibody that's there, and you go into this decline phase. So we have these 4 phases, plateau and decline. Now, all right, what happens when we see this a second time? When we see this antigen a second time, and what I've done here is I've put on the first primary response here again so you can distinguish the primary and secondary response. There is a lag phase in the secondary response, but it's much shorter. Now, instead of taking a week to 10 days, it's going to take just 3 or 4 days. It's going to be a much shorter lag phase. There is a log phase, but now the titers of antibody that you achieve are much, much higher than you get after a primary response. So you get much higher titers of antibody. There is a plateau phase eventually, because even after the secondary response, those plasma cells are going to die off. And so the rate of synthesis equals rate of decay. And then you go into this decline phase. But now the decline phase is very, very gradual. These antibodies will persist for weeks, months, some years, sometimes even a lifetime. So there's a very, very slow decrease in the amount of antibodies in this decline phase. Now, there's a high degree of specificity not only in the antibodies that are produced, but also in generating this secondary response. Only exposure to the same antigen a second time will give you this response. In most cases, even if you're exposed to another antigen that is very, very similar to the first antigen, you don't get this memory response. You get another primary response. All right. So this is called the memory response. You'll also see it referred to as the anamnestic response. It comes from the Greek meaning memory. So to get this secondary response, you really have to be exposed to the same antigen a second time, and then you see the secondary response. There are a few exceptions to that rule, but by and large, that holds true. All right. That's what's happening to the kinetics of the response. In addition to the kinetic changes, there are differences, there are qualitative differences in the primary versus the secondary response. So one thing, there's class variation. Now, instead of just looking at total antibody, what I've done here, the total antibody is in red. This is what you just saw. What we're going to look at is ask, well, what's the different classes of antibodies that are being produced? And what you see, after the primary response, the main antibody that is being made is IgM antibody. There is a little bit of IgG made later on in the response, but the prime antibody, the characteristic antibody of the primary response is IgM antibody. In contrast, in the secondary response, while you do see some IgM being produced, the bulk of the response is in fact an IgG response. And when I say IgG, that could be IgE or IgA as well, if it switches over to making one of those classes of immunoglobulins. So they would follow this blue line just as well as if it was if the switch went to IgM or IgA or IgE. All right, so the characteristic antibody of the primary response is IgM antibody. The characteristic antibody of the secondary response is IgG. And it's, in fact, the IgG response that accounts for this huge increase in titer and this very slow decline in the antibody concentrations. So there's a change in the class of the antibody that's made. There's also a change in the affinity of the antibodies. The affinity tends to, in the secondary response, increases, and it's particularly noticeable when there's a low dose of antigen. So what's plotted here is the affinity days after immunization. Here's after the primary response, here's after the secondary response. And you can see after the primary response, there's a slight increase in affinity of the IgG antibodies. But after a secondary response, you get a nice increase in affinity, particularly when the dose of antigen is low. If you look at the affinity of the IgM antibodies, they don't change. Right? So it's only the IgG. And again, when I say IgG, that could be IgE or IgA. The same thing applies to IgE or IgA. There's this increase in affinity. This is referred to as affinity maturation of the antibodies. And there's two reasons to explain why you see this affinity maturation, particularly with the low doses of antigen. So let's illustrate that. Suppose we had a series of B cells that all could react with a particular antigen. but they reacted with that antigen with different affinities. And let's say the affinities can range over 10 to the 5 up to 10 to the 10th. When you have a high dose of antigen, all those clones will be stimulated. There's enough antigen so that every clone, even the low affinity clones, are going to be able to have antigen to bind to them and get activated. So they will produce antibodies of the same affinity as the cell surface immunoglobulin, and you end up with a population of antibodies that have sort of a moderate affinity because it's a mixture of antibodies. But when the antigen concentration is low, you preferentially stimulate or activate the high affinity clones. So only those clones are the ones that are going to be activated, and so you end up with antibodies of a higher affinity. So that's one of the reasons, that particularly with the low dose of antigen. The other reason why you get affinity maturation is due to somatic mutation. Because upon secondary exposure to the antigen, the somatic mutation mechanism is activated, and we begin to introduce mutations in the immunoglobulin molecules. Any mutation that results in an increase in the affinity of the antibodies will be selected for, particularly when the concentration of antigen is low. So those mutations that result in an increase in affinity have a selective advantage, and those are the clones that are going to be activated by the antigen. Any mutation that results in a decrease in affinity will be selected against. All right? So what you do, the immune system, by turning on the somatic mutation mechanism, is fine-tuning the response so that you select for higher affinity antibodies. Okay? So the antigen is not directing which mutations are occurring. The mutations occur randomly. Some of them are going to be the higher affinity, some of them are going to be lower affinity. Only the higher affinity ones will then be selected out. Okay? So that's why we get this affinity maturation. There is also an increase in the avidity then as in the secondary response, and that's not too surprising. If the affinity is going up, the avidity is going to go up as well. Okay? So you'll see in the secondary response we see class variation, affinity maturation, and avidity increases. Now what's less obvious is that there's also an increase in cross-reactivity. And the better way of probably saying that is that there's an increase in detectable cross-reactions. And let me illustrate that. Suppose we immunize with an antigen. And let's say that in the primary or early in the response, you get antibodies of a certain affinity. We'll call it 10 to the 6. Later on in the response, because of affinity maturation, the antibody affinity has increased roughly about 1,000-fold, okay, after a secondary response. And let's say that there is a cross-reacting antigen that would cross-react with these antibodies. But they do so with 1,000-fold lower affinity, okay? So early in the response, the cross-reactive antigen would be reacting with an affinity of 10 to the 3. Later on, it's going to be reacting with an affinity of 10 to the 6. Now, in order to detect an antibody response, or an antigen-antibody reaction, remember we said we had to have a certain affinity in order for the thing to stick in, the antigen to stick tightly into the antibody combining site. So there's a threshold affinity above which you will detect, below which you will not detect. So let's say that that's about 10 to the 5. Well, obviously, then this would be a detectable, but this would not be detectable. But now, when the affinities have increased because due to affinity maturation, they're both actually detectable. So as a consequence of increasing affinity of the antibodies, you also get an increase in the cross-reactivity of the antibodies that are produced. Okay, so those are the changes, the qualitative changes and the quantitative or kinetic changes that distinguish primary from secondary responses. Now let's try to go back through that response and explain what's happening at the cellular level. Again, we're still dealing with T-dependent antigens. All right, what's happening in the lag phase? Well, obviously, what's happening in the lag phase is clonal selection. Those cells that have the appropriate receptor for antigen will bind the antigen, and they will get activated. Now, this is a T-dependent antigen, so they're going to require some T-cell help, and you'll find out what the nature of that help is. when Dr. McKellop gives his lectures. But eventually these cells are going to be in this lag phase. They're going to be activated. They're going to begin proliferating, increasing that clone of cells. And then some of these cells are going to start going in their terminal stages of differentiation towards plasma cells and then ultimately beginning to secrete antibody. So when they get to that point where they start secreting antibody, we enter into the log phase. And so these cells are now churning out antibody. They're secreting lots of antibody. But it's primarily IgM because IgM is the first constant region that is next to that rearranged VDJ. So that's the first thing that it can express. So it expresses IgM antibodies. Now, eventually, as the response goes on, some of those cells switch to making other classes of immunoglobulin, IgG or IgA or IgE. So a few of those cells will switch over to start making IgG, and so you do see a little bit of IgG antibodies produced in the primary response. Now, eventually, though, as these plasma cells start dying off, no more antibody is going to be made, and the rate of synthesis equals the rate of decay, and you enter into the stationary or plateau phase, and then you go into this decline because all these cells start dying off, and they're not synthesizing antibody anymore. But not all the cells die off. Some of these cells actually get reserved into a pool of cells referred to as the memory cell pool. And so what you have after primary exposure, you have an increased pool of memory cells. And these cells are the cells that are going to be available then for secondary exposure or upon secondary exposure to the antibody. So you reserve some of these cells in this memory cell pool. So after a secondary exposure to the antigen, what you see in the lag phase, yes, there are some virgin or naive B cells, cells that have never encountered antigen before that have been generated. So these cells can be stimulated, and yes, you can get some production of IgM from those cells. But the bulk of the response in the secondary response is not coming from those virgin or naive B cells. They're coming from that memory cell pool that's there after the primary exposure. So the bulk of the response is coming from these cells, and if the bulk of the response then is in an IgG or an IgA or an IgE response. So because of this increased pool size, we get a lot more of this antibody produced as well. Okay, but eventually then again, the plasma cells start dying off, and synthesis equals decay, and then you go into this decline phase. But now the decline phase is very gradual, because some of these memory cells actually can continue to just secrete low levels of immunoglobulin. They're not plasma cells, they're B cells, but they can secrete low levels of immunoglobulin, and you have this sustained long production of antibodies that may last for weeks or months or even years. Now, we said that this was a T-dependent antigen, so I do want to just talk a little bit about the T cells, and I'm just showing you what's happening after a secondary exposure to the T cells. The same story that I just said for B cells also happens to T cells. And you get, after the primary exposure, some of the T cells are reserved as in a memory cell pool. So after secondary exposure, yes, you can get some virgin T cells activated, but the bulk of the response is coming from these memory T cells, which differentiate into T helper cells. And one of the things these T helper cells do is then they secrete cytokines that help the B cells differentiate, proliferate, and so on. So they also generate an expanded pool of memory cells. And in fact, the memory cells are, in the T cell population, are actually longer lived than the B cell memory. B cell memory dies off a lot sooner than the T cell memory. T cell memory is very long-lived memory. And so if you haven't been exposed to the antigen for years, the secondary exposure doesn't occur until years after the primary exposure. It's primarily the memory T cells that are responsible for the secondary response, because they're ready to go. They immediately help the B cells. The B cells will switch a lot sooner to making IgG, and these are the ones that are responsible for really long-term memory, the T cell memory. All right, that's T-dependent antigens. What about the T-independent antigens? Well, you see all four phases after exposure to a T-independent antigen. There's two differences. One is that the only IgM antibodies are made. You don't switch to a different class of antibodies. There's only IgM antibodies made. And the second feature is there's no secondary or anamnestic response. Secondary exposure to a T-independent antigen just gives you another primary response. So the difference with T-independent antigen is no memory, IgM antibodies. Okay? All right. So we said that in the case of these T-dependent antigens, that you get this switching to other classes of antibodies. So what we need to talk about now is how that switch occurs. And again, the answer came from an analysis of the immunoglobulin genes. So we go back to the genes and see what's going on. All right. So here's the rearranged VDJ. Here's the constant region heavy chain genes. Okay? What happens during class switching? When a cell switches from making IgM, the first thing it can make, of course, is IgM. When it switches to make another class, yet another DNA rearrangement occurs. All right? But this is different from the rearrangement that generated the VDJ. This does not, for example, use those RAG genes at all, the RAG1 or RAG2. It's a completely different type of rearrangement that's going on. But it is a DNA rearrangement. And if you look at the genes, what you see is up front of all of these constant region genes, with the exception of delta, all of these constant region genes have a signal in this intron up front of the upstream of the gene, which is called a switch signal. All right, that's why it's labeled S. So there's a switch signal. So what happens during class switching is that a recombination event occurs between the switch signal in front of the mu heavy chain, mu constant region gene, to one of these other constant region genes. So in this example, we're bringing it switches occurring between this switch site and this switch site. So this will bring it next, then we'll be using the epsilon gene. So what happens after the switch has occurred, where first of all you can make this transcript, now you delete out this piece of DNA. The VDJ is now associated with the epsilon gene. So now you transcribe this into a primary transcript that encodes an epsilon heavy chain. Okay? Now, notice, even though you have switched to a different class of antibody, the specificity will remain the same. Because it's the same VDJ that's going to be used. And when this epsilon chain, heavy chain, associates with whatever light chain that cell is making, you will express the same specificity that was present on the IgM molecules. And that's why cells, when they switch, begin to secrete antibody of a different class, but of the same specificity. The B cells don't make antibodies of different specificities after the switch. They keep the specificity the same. And what's the advantage of that, again? Why would the cell want to switch to a different class? What advantage is it? It changes the effector functions. You can keep the same specificity but change the effector functions, okay? So this would now have this would be an IgE molecule that's being made. Now, this switch requires T cell cytokines. And in fact, the type of cytokines that it's exposed to will determine which class of immunoglobulin it's going to switch to. So the T cells help decide then which class of immunoglobulin the cell is going to switch to. All right, so some ways may cause it to switch to E, some may cause it to switch to one of the gamma chains, and so on. All right, any questions about the switching? All right, one of the things that I think you can see here is that if a cell switches, say, from making IgM, if it uses, say, goes to gamma 3, which is the first constant region here, if the cell switches to gamma 3, then in theory could switch again to one of these other classes, right? of immunoglobulin, because if it switched first to gamma 3, it could later on then switch to gamma 1 or gamma 2 or epsilon and so on. And in fact, you can see that as well, that that you can get subsequent switches to other classes of immunoglobulin. However, once you switch to alpha 2, there isn't anything else to switch to. That's an endpoint, right? Because that's the last one. Everything, all this other, anytime you make a switch, remember, you're deleting out the DNA in between there. So once it switches to the last constant region, then it's not going to be able to switch again. Lyle?
So the idea that you can't have switching to a delta?
Well, again, delta is handled differently. Delta is handled by expression of delta is done by differential processing of that message, the primary transcript, okay? So when we talk about class switching, you're not talking about making a delta, because delta is handled different. Class switching refers to switching to any of the other classes of immunoglobulin with the exception of delta. There was another question here? Okay. All right. And then, but again, keep in mind, the specificity is never going to change in this process. Okay. The last thing I want to cover is membrane versus secreted immunoglobulin. If you remember, we said that IgM, or the mature B cells, have on their surface these two classes of immunoglobulin, IgM and IgD, that act as the receptor for antigen. And we also said that cells, after the exposure to antigen, begin to secrete antibody. And they secrete antibody the same specificity. So how does a cell go about doing that? So how does a cell then determine whether or not an immunoglobulin molecule is going to be destined for the surface of that cell and become a surface immunoglobulin versus one that's secreted? And again, the answer comes from an analysis of the genes. All right, so let me orient you here. What we're looking at is we're looking at the end of the C mu gene, or let's say that this is the C mu gene because there's this extra exon. Remember I said that each of the domains of the molecule were encoded for by a different exon. So here's the CH3 domain of the mu chain, and here's the CH4 domain. It's one of these that has that extra domain, okay? Now, all the VDJ is, of course, out here, and all the other heavy chains are out there someplace, the constant region genes, okay? So we're just looking at the end of the C mu gene. If you look in the intron, then after the last the CH4 exon, there are actually two additional coding sequences that are referred to as the M coding sequences. And those exons code for that extra 20 or 30 amino acids that make up that tail piece. Right? They actually encode that tail piece. Now, also, in the introns between these coding sequences, right there and right there, there are two potential polyadenylation sites. All right, so what happens when this gene is being transcribed into its primary transcript? There's two different kinds of messages that we can make from this. If this polyadenylation site is used, and when the transcript is being made, poly A is inserted in response to that signal for polyadenylation, you get this kind of a message that ends like that. When that is processed then to remove all these introns, and we're moving out all the various introns to bring all the coding sequences together, you end up with this type of a message, which is then a message for the secreted form of the antibody because it doesn't have the tailpiece exons. Those have not been expressed in this message. So this message would encode for an antibody that's going to be secreted. If, on the other hand, as you go through this, if you skip this polyadenylation site and use this polyadenylation site, then what happens is then we end up with a primary transcript that looks like that, and this then gets processed by removing out all these introns, and we end up then with a message that encodes now for the amino acids in that tail piece. This then, the tail piece, is the instructions that say, okay, put me on the surface of the cells. And this goes through a different vesicular pathway that goes to the surface of the cell. And this immunoglobulin is expressed on the surface of the cell. So the cell can express either a secreted or a membrane-bound form. In the B cell prior to, mature B cell prior to being exposed to antigen, this is the processing that occurs. This polyadenylation site is used. After exposure to antigen, this polyadenylation site is used, and you start secreting the antibody instead of putting it on your surface, putting it on the surface of the cell. Now, and notice, even though the cell is making can make these two different forms of the antibody, a secreted form or a membrane-bound form, the specificity hasn't changed. The same VDJ that was expressed on this membrane form is also going to be used for this secreted form. So the specificity of the antibodies don't change. And that's why when a cell secretes the antibody that's secreted will bind to the exact same antigen as the immunoglobulin that was on the surface of the naive B cell that got exposed to that antigen. Okay? Specificity doesn't change at all. All right? Now, each of the heavy chain genes, the constant region genes, every one of those heavy chain constant region genes has these membrane exons associated with it. So what that means is that after switching to a different class of immunoglobulin, after the class switch has occurred, those cells then can either have a secreted form or a membrane-bound form. And so after exposure to antigen, if you look for cells, for B cells, you will find B cells that actually can express on their surface IgG or IgA or IgE. But those are cells that have first been exposed to antigen because they underwent the class switch. Okay? So later on, after class switching has occurred, you can also then begin to see cells that are expressing other classes of immunoglobulin on their surface because every one of the constant region genes has those membrane exons associated with that code for that tail piece. Okay? All right. Any questions about that? Yes. No. What I'm saying is that prior to exposure to antigen, B cells, the naive or virgin B cells, just express on their surface IgM and IgD. So they are using this site, okay, to put it on their surface. After exposure to antigen, even in a primary response, after exposure to antigen, this site will be used and the IgM will be secreted, all right? Once class switching occurs, then you can start making IgG that is secreted. But some of the cells actually can then put it on their surface as well because they can use these membrane exons as well. Yes? No, not necessarily. But remember, if this site is used, this will happen, right? But this is occurring during transcription. So you can transcribe this and then on a different transcript use that site, right? Because this is the transcript down here. So as you're making it, you either polyadenylate at this site or at that site.